Cholesterol helps bacterial toxins kill cells. Researchers find cholesterol helps bacterial toxins kill cells. Cholesterol stabilizes and binds the toxin protein to the cell membrane. Researchers at the Indian Institute of Science IISc Bengaluru have found that cholesterol present in cell membrane plays a crucial role in stabilizing and binding together the pore-forming toxin cytokinin A. The pore-forming toxins form the largest class of bacterial proteins causing virulence that kills human cells. The cytokinin toxin is secreted by E. coli, Shigella, and Salmonella. The toxin secreted by E. coli bacteria is water-soluble and binds to the cell membrane. The binding of the water-soluble toxin to the cell membrane does not depend on cholesterol present in the membrane surface. In fact, the binding drops in cholesterol-containing membranes. Binding is only a part of the pore-forming process, and cholesterol has no role to play, says Dr. Rahul Roy from the Department of Chemical Engineering at IISc, who led the team. Toxin movement. Once the toxin gets bound to the cell membrane, it does not stay in one place. Instead, it keeps moving around the cell membrane surface. Using a powerful microscope that allows them to look at single molecules tagged with a fluorescent tag, the researchers could actually see the toxin proteins moving around. Unlike the usually observed Brownian movement, these proteins tend to move around fast and then slow down before picking up speed and moving fast again. This happens even when no cholesterol is present. The structure capable of puncturing the cell membrane is supposed to slow down the protein movement. So this suggested that the structure of the bound protein is similar to the water-soluble protein and different from the structure that pierces the cell membrane. Cholesterol interaction. In the presence of cholesterol, the protein stops moving quickly. Using molecular dynamic simulations, we found cholesterol interacting with the protein just as we suspected. The interaction was with the region on the protein that is responsible for forming the pore, says Dr. Roy. Binding to cholesterol per southeast does not stop the motion of the protein, but on binding to cholesterol, the structure of the protein undergoes a change resulting in slowing down of the motion. The change in the speed of motion happens even in the absence of cholesterol due to the change in structure of the protein. But in the absence of cholesterol, the protein is unable to maintain the structure required for pore formation. The structure of the toxin is stabilized in the presence of cholesterol, and that is essential for pore formation, says Pradeep Satyanarayana from the Center for Biosystems Science and Engineering at IISc, and first author of a paper published in the Proceedings of the National Academy of Sciences. This is a clever strategy by the bacterial to use the toxin to specifically target only human/animal cells, while the bacterial themselves are protected from the toxicity since cholesterol is absent in bacterial membranes. To be able to rupture the cell membrane, the proteins bound to cholesterol have to come together to form a ring-like structure comprising 12 molecules. The coming together of the molecules to form a ring-like structure is also enhanced in the presence of cholesterol. Computer simulations showed that when two pore-forming protein molecules come together, there is a small pocket where the cholesterol goes and interacts with the proteins. So cholesterol provides additional support to hold the two molecules together, says Dr. Roy. Studies by other groups have shown that cancer cells in mice can be reduced dramatically by using cytokinin toxin. Based on our study, we can work on making the toxin target only the cancer cells. We can also use cholesterol-like molecules to prevent the toxin protein from changing its structure, thereby prevent cell destruction.